right. Well, if we're all set up and ready to go, then in that case, if we're all ready, I'm going to go ahead and call to order the 1250th regular meeting of the ESW. Thanks to all in attendance, both in person and virtually. First order of business, we'll have Recording Secretary Gary Hubble read the January 2025 minutes. Take it away, Gary.
Thank you, Andrew, wherever you are. The 49th regular meeting of the Entomological Society of Washington convened shortly after 7 p.m. on January the 16th, 2025, in room WG32 in the National Museum of Natural History, with President Andrew Johnson greeting members, visitors. President Johnson was himself occurring in a virtual format. 12 members, 6 visitors, and 25 virtual participants were in attendance. At the meeting, at the request of President Johnson, Recording Secretary Gary Hubble read the minutes of the December 2024 meeting, and these were approved by the audience. Webmaster Lourdes Chamorro offered a detailed webmaster report registering activities of the ESW website between 15th of December 2022 and the 16th of January 2025. Your report reflected 9,354 views, of which were mostly from the United States, with other major views from Mexico, Canada, China, and Japan. She also noted that society merchandise is soon being offered for sale online, with current items including drinking mugs and sweaters. Membership and Communications Officer Matt Buffington enthusiastically presented the names of the following membership applicants for the society: Roy Beckemeyer, Raymond Sherbo, David Frischtak, Jasper Pickman, Stephen Gamare, Vince Brack, Zachary Leahy, Jason Williams, Jeffrey Schultz, Kathleen Evans, and Renan Carenho. Past President Don Weber and President Andrew Johnson both thanked Treasurer Abby Kula for her devoted attention to the conversion of the society to the status of a District of Columbia nonprofit corporation. Program Chair Alan Norbaum introduced past President Don Weber, who presented the topic Adventures with Vegetable Pests, Behavior, Control, and Cereal Chemicals. In pursuit of the control of various pest species, Don has engaged in research of chemicals that influence the successful accumulation and reproduction of pest species. Male-produced aggregation pheromones are powerful tools, and there are steps in recognition and utilization of these chemicals. Enumerated stink bugs, BMB, produce a chemical, bergantiol, which is commercially available. This chemical attracts thousands of females and can be used in all seasons. Several other pest species were outlined in presentation, including striped cucumber beetles, harlequin bugs, leaf-footed bugs, and Colorado potato beetles. With these species, populations were field attracted to various levels of mixes of pheromones and other chemicals. Throughout the research, it was determined that the use of chemicals must be coupled with biologies for better results. The meeting was adjourned at 8:25 p.m.
Thank you for the report. Do I have a motion to approve the minutes?
Approved. Second.
Then, having heard a motion and a second, the minutes are hereby approved. So on to the next order of business. We are still working on putting together the audit committee and audit. I know it's late. I apologize for that. The last Couple of weeks, as some of you may know, have been a bit trying for those of us in the federal civil service. So I appreciate your patience very much while we work through to the audit. With that, I'll turn over the floor to any officers who have reports for this evening. Would anyone like to take the floor?
Yeah, I have something to say real quick. We're still deciding on final prices, but we're going to do from now on an in-person special sale for a lot of our materials that we offer on the website shop. But you guys will get a pretty significant little discount. So be sure to check out the little cart we have over here after the meeting. And then also we have free material as well.
Don't confuse what's free and what's.
Oh, no, I'll be over there.
So let us not confuse. Oh, Don pointed out this could be an incentive for in-person attendance. Thank you, Don. Anybody else for officer reports? All right. Hearing none. Are there any new members or visitors who would like to introduce themselves either in person or virtually? We'll start with anybody in person. Any new members or visitors?
We have a couple of visitors.
Yes.
Today.
David. I go to virtual. I like bugs, I guess. Yeah. Place. Yeah, weevils. So, yeah.
Hi, everyone.
Welcome, welcome.
No, not really. Weevils.
And I'm David's mom, Patricia.
Thanks for coming. All right. Anybody else in the audience?
Hi. Thank you for the invitation. I'm Mariela from Argentina. I'm starting a postdoc with Hannah Wood, and I will be working with spiders. Also in functional morphology, of something of behavior. So I will be starting to come to this intervals.
I'm Lais. I'm Brazilian. I am a PhD student at George Washington University. I work with army ants. I study the evolution and development of army ants. And I'm here as a grad fellow in Ted Schutz's lab. So my advisor from school is Scott Alper.
Welcome to the both of you. Thanks for joining. Anybody else in person, or how about virtually?
My name is Zachary Briepino. I have just begun as a postdoc under Mike Gates in the Systematic Technology Laboratory, working on identification tools for pine sawflies, mostly indigenous Cyprian.
Glad to have you.
Okay. Hi, everybody. My name is Morgan Thompson. I am a new postdoc with Karen Burkhardt at the University of Maryland, and working with her in collaboration with folks at the Smithsonian Environmental Research Center on the biodiversity tree experiment, if you guys are familiar with that.
Right. Thanks for coming.
Happy to be here.
All right. Anybody else in person before we open it up to Zoom land?
Hi, if you can hear me.
Can hear you.
Yeah, this is Chuck Sexton. I'm a retired ecologist in Austin, Texas. Studied and worked with birds, endangered species of birds most of my career, but I've had an interest in moth and moth identification over the last 20 years. So I'm happy to be at my first ESW meeting.
Well, thanks for coming. We're glad to have you.
Hi, can you hear me?
We can hear you.
My name's Freddie Purnell. I'm a graduate student at the University of Pennsylvania studying the gene regulatory and epigenetic factors that establish and maintain caste identity in the leafcutter ants Atacephalotes. Glad to be here. I always like bugs. My friend told me to come here. He comes here all the time, so now I'm here.
All right. Glad to have you. We have one in the chat. So we have I'm really sorry if I hope I'm not mispronouncing this. Solanga Akimana. So I am Solanga, PhD student at Drexel University, Philadelphia, working on systematics of crane flies with Dr. John Gelhaus as my advisor. Thanks for joining. And is there another one down there? All right, go for it.
Hi, everyone. I'm Jorge Bustamante. I just started a second postdoc at the USDA in IIBBL, so in the Lee lab. I'll be studying tick behavior, tick repellency, tick feeding behavior. And yeah, former biomechanist, studied hawk moths, did a bout with urban entomology with bed bugs. I see Harold Harlan in the chat. That's awesome. And yeah, it's looking forward to meeting everyone.
Glad to have another biomechanics person, and as always, it's good to have the arachnology people come and join as well. Anybody else out in Zoom land? Here we have a comment in the chat. Lily Fetterman, one of John and Solong's students at Drexel. Welcome, Lily. Thanks for joining.
Andrew, I could say something too.
Go for it.
Go for it. Gary Humphrey here. I'm a Myrmeco statistician at the University of Guelph in Canada. A Myrmeco statistician is, nobody needs to know what a myrmecologist is, but I am actually employed in the Department of Mathematics and Statistics as a statistician, but I love ants, and I want to give a shout out to Ted Schultz, who I met first time when he was still a PhD student at Cornell. Got to do a few research trips to the Smithsonian in the good old days, where you could just wander through the place at night. And I did give a talk at the ESW Entomological Society of Washington many years ago, but I'm not sure how many years ago. It was a long time ago, and it was absolutely a delight to be down there. We always, as a Canadian up here, I wish your scientific community well, and we're always thinking of you.
Thank you. No, we thank you for that, and welcome back, Gary. Welcome back. We have one in the chat.
Oh, I'll mention one thing. I have office hours, online office hours at 8 o'clock. If I have to leave beforehand, just before that, you'll know why.
All right.
Okay, thank you.
Freddie Purnell says that his advisor, Shelley Berger, is also here as well, or maybe that he's in that lab. One or the other. Well, in any case, thanks all, and welcome. This should hopefully be a very entertaining evening with an excellent presentation coming up. If there are no more new members or visitors, I'll briefly open the floor. Is there any unfinished business or new business, either from the last meeting or that needs to be introduced during this meeting that has not already been covered?
There, he gave you the floor.
We have one more person in person.
Can you hear?
Can you hear? Can you hear us?
Yes.
I'd like to introduce my pal, Sean Reagan from Riverside, California. He's married to a very good friend of mine, and I found him here wandering around DC doing Capitol Hill stuff. And I said, hey, you should come to visit in Saco, Washington, and come visit a 100-year-old entomological society. So here he is.
Hi.
Thanks for coming.
Thank you. I'm a rolling
serendipitous.
So this fits.
Correction, 150 years old.
150.
And I have a note here. This is the October 2023 issue of the proceedings, and it's the last one in the library. With 4 issues printed each year, why are we 5 issues behind in publications? Where's
Carrie. Where's Carrie?
where's our curator?
The editor.
Do we know why?
Oh,
Although we've been this far behind before, but this one is very thin
We're supposed
too.
to get caught up.
Hmm.
I don't think it's just been populated in the library yet. I think we're only a month behind.
I was gonna say, I've received at least 2 mail copies of recent issues of the proceedings of the ESW.
Oh, good, good, good. So it's just the library that's behind
Yeah,
for some
yeah.
reason.
Yeah, physical.
Oh, good. I was worried.
Right.
Because all, the more recent issues I actually have in my office, so that's probably.
Okay.
Sorry about that.
Well, we looked around in the office there, but it's so full
No,
of
it's
stuff.
not in- I have the very most recent one in my office.
Your office, not the library.
Yeah, yeah.
Okay. Well, as a former ESW membership secretary, our membership is global, and all libraries should have printed copies globally, so.
Not being familiar with the library, or overly familiar with the library, I should say, is there any particular reason that they would be like excessively behind in getting copies of ESW proceedings?
Talked to the librarian today, and he wasn't aware of any being behind on any copies, but
I
that.
don't know what the distribution cycle is like, but maybe I'll make a note of that one and see what.
Yeah, I can rectify that.
The curator says she can rectify this.
That's an easy fix. I can move a few into that for each,
Good.
for each issue.
Yeah, yeah, thank you.
Make it one. We'll bring that one. We'll talk about that at the executive. Let's see. Okay. Is there any additional new or unfinished business before we move on to presentation of specimens? Okay. Hearing none, does anybody have any special notes that they would like to present or any excellent specimens to exhibit to the group?
I have several.
All right. All right. Take it away.
Many exhibits because I kept forgetting. The first is a unique ant discovery. This follows an exhibition by David Firth in 2007, where he exhibited a alarm clock radio full of Tapanoma ants. I have here Staples clear glue that was found on our daughter's floor in her bedroom, full of Tapanoma sessile, which I've never seen that before. They've gotten themselves into a sticky situation. So I'll pass this around. I thought that was kind of because new kind of ant trap, and because it's an ant theme talk tonight, it makes sense. I also have some artifacts here from the International Congress was in Japan this last year, and we've got several things that Lourdes and I brought back. One are these insect-shaped candies that my colleague Tatsui Ide made as like a side project. He's a gall wasp worker, but also enjoys insect art and came up with insect-shaped candies. Okay, don't open it. I don't know what flavor they are. It's only to look at. Have you ever been accosted collecting insects by people wanting to know, what are you doing? Well, all you need is one of these wristbands. Of course, it's in Japanese, but what it says is, trust me, I know what I'm doing. I'm an entomologist. So I have an orange one. Lourdes has the green one. Don't open it and try it on. Just leave it in the package.
Is that another potential product for us to?
Hey, I like how you're thinking that. In English.
Maybe Spanish.
In Spanish too. That's a great idea.
Southern.
Southern.
They have a cute little honeybees down there.
So as a hymenopterist working for USDA, it was part of my job to do the identifications of Vespa mandarini, the so-called murder hornet. This is obviously a species that Japan deals with like on a regular basis. And I picked up this really great guide to Vespids of Japan. And it actually has some incredible diagrammatic keys, like flowcharts, telling all the different species apart, which I might mine for my own hornet identification guide I'm working on right now.
So is the page for that one just like one giant image?
Does it look like this? And this one, Lourdes, do you want to introduce this one? Oh, okay. So this is a book of amazing beetles signed by the author. And this is a colleague of Lourdes's. And it's just a beautiful photographic guide to, are they beetles of Japan?
Yes, but he also works on beetles associated with ants.
Oh, that's right. That's right.
There's a theme going on.
Yeah, that's right. I think there's one on the back, if I'm not mistaken. So, yeah. Those are all my, oh, just as a last thing. So going into the books, the ones on the left are from the late Dave Smith. They were in his office. They have not been claimed by anyone, and they are free to anyone who wants to take them to a new home. Don't neglect the Saricids of North America guide. It's in color. Thanks, everyone.
Fred's asking, is there any way for the people out in Zoom land to see any of these things? Might be a bit difficult. I don't know. Short of holding them up to the camera. That's a good point, though. Do we have access to a like a handheld webcam or something? That might be something to.
Thought about it, but I don't think we do. We can just hold it up to the camera here, I think.
You could turn on the camera on the left too and hold it up. That might be a little-
So we'll do some online photography and send it back. Okay, here's the wristband. Or armband. Trust me, I'm an entomologist. And there's instructions on the back. Here are the candies. Like various little like spiders and stuff like that. Oh, no, I'm totally wrong. They're actually galls. They're actually little candy galls, which makes it even more hyper specialized. So these are candy galls. Sorry about that.
I'm sorry.
It's actually even cooler than I realized. Okay, here comes the Tapenoma sessile staple glue. Yeah, there's at least 400 of them.
What do you think they're attracted to in that glue?
Oh, did we hold this one?
Here's the
Okay, anybody else?
that's the tan guy. Oh, here's the.
Anyone not seeing the lovely ants?
Okay, that's all.
Very cool.
So the cats just wandered in there on their own. They liked it.
It was something attractive.
Interesting.
It was open just a little bit, so they actually went in through the.
What a way to go.
Yeah.
I think
So, ladies and gentlemen, it is now nearly 7:30. I'm calling it to order. We are, I think, at the appropriate time for our speaker. Thanks for the presentation of specimens, everyone. I will now let Alan introduce our speaker, Dr. Evan Economo.
Before I do that, first off, Gary, we have 25 people on Zoom.
Okay.
Our upcoming speakers are another scientist from the University of Maryland who happens to be with us tonight, Morgan Thompson, will be next month. And then we have our own Jeffrey Sosa Calvo, who's also here tonight. And in May, we have Jerry Cook. So have the pleasure to introduce Evan Economo, our speaker tonight. He is the Gahan Professor and Department Chair of Entomology at the University of Maryland. He received his PhD from the University of Texas at Austin. He completed a postdoctoral fellowship at the University of Michigan and was professor at the Okinawa Institute of Science and Technology in Japan. For 12 years before he moved to Maryland last year. His research group aims to understand what generates and regulates biodiversity across levels of organization while working to document the vast diversity of the planet. They specialize in integrating biodiversity knowledge from perspectives including natural history, biodiversity collections, genomics, biogeography, and 3D imaging. I have a particular focus on ants.
Yeah, thank you very much. Let's try to. Okay. So thank you very much, and thank you for the kind invitation and introduction. I didn't know we were going to be sharing cool Japanese entomological things, but next time maybe I can bring some videos of being on Japanese TV talking about insects. It's really great country for entomophilia for sure. Yeah, and so also great to be here. I moved to the University of Maryland in September, and I'm still connecting with all the local entomologists here in the DMV. So I'm really looking forward to interacting with all of you more. And don't be a stranger if you're ever up at our department. Stop by and say hi. And yeah, so today I'm going to be talking about morphology, and this is, of course, I'm going to zoom out a little bit, a very old preoccupation of people who study entomology. And it goes back to some of the real early anatomists. One of them here, Sir John Lubbock, who's actually a really interesting guy. He was an MP in Parliament and just a real Renaissance man. He made a big impact in anthropology, but also liked to dissect ants and document all the muscles inside ants, and did really incredible drawings and anatomical studies with very rudimentary equipment at that time. And now we're kind of in the era of big data. And I think if I were to try to describe what we're trying to do in my own lab over the years, it's sort of generate big data on different aspects of biodiversity and integrate it. And I sort of think of this as the bedrock data that we're going to build our science on in the future. The geographic distribution of every species, the phenotype of species, the phylogeny and systematic framework that we use to organize life, and genomics. And you could probably add a few other things in there too. Is how can we attack and complete the massive challenge of documenting biodiversity across all these dimensions. And it's a big challenge, but we also have a lot of new exciting technology and methods to try to go at it. But today I'm really going to talk about work we've done on quantifying phenotypes and the evolution of phenotypes. And even though when we think of big data in biology, we usually think of genomics and all this kind of different molecular methods, we also have analogous technologies that are generating big data on morphology and anatomy. And many of you probably have seen this before, but micro-CT is one such method, a method that we use a lot.
And micro CT is basically like a medical CT scan, but with very, very high resolution. So a CT scan for a human being, you get in the hospital has a resolution of about 1 millimeter. So it's good for a human, but not so good for an ant, right? And so the resolution of a micro CT is that we would use is less than a micron. So we would put the specimen inside here. There's an X-ray source that shoots an X-ray while the specimen rotates and gets a flat, X-ray image of the specimen in all different directions, and then computationally reconstructs a 3D image or 3D volume. And of course, I'm going to talk about ants, and I just want to give sort of some context of what is interesting to us and motivates us about the phenotypes of ants. And ants are one of the most ecologically dominant terrestrial animal groups in terms of biomass, in terms of their interactions with other organisms. Really, one of the groups that dominates the terrestrial environments on Earth. And the keys we normally talk about to their success are, of course, their sociality, their division of labor, their communication, their ecological and behavioral flexibility, but also the wingless ant worker phenotype. So actually, that's one of the key innovations of ants is the loss of flight. Most of you probably know, the ancestors of ants was a winged insect, a winged wasp. And one of the key innovations of ants was actually to have a caste that lost flight. And that allowed for a broad reorganization of the phenotype. So changes to the thorax of the ant or the mesostoma. You see this is actually an ant queen or a flying insect. The inside is really dominated by the wing muscles and the constraints that it needs to achieve in order to fly. But inside ants, you see without that constraint of needing those wing muscles, you see real reorganization of the endoskeleton and the different muscles and arrangements that can optimize the worker for ground labor. And likewise, the head, many other hymenopterans are hypognathous. Ants are prognathous, meaning their mouth looks forward instead of down. And that allowed for many different changes and reorganization of the head, and also diversification of the mandibles. So in our lab, we've been working over the years on many different aspects of the evolution of morphology, everything from locomotory apparatus to the brain, to the stinger and other things. But one of the things we've really been interested in is mandibles. And that's what I'm going to talk about for the rest of today. So when you look at ants, the mandible diversity is really off the charts compared to other insect groups. It's one of the most striking things about ants. You can see a bit of that diversity there. And this is another look at that diversity. These are CT scans across hundreds of different species of ants. And my student, Julian, who is doing his PhD on the evolution of mandible morphology, has taken them out in a range of them. You can see the broad diversity of different shapes you get with ants. And actually, this is a PCA, so it gives a morphospace of different ant mandibles. And you can see that, I don't know if you can see very well, but that back shape is the predatory ants. Many of those really weird, unusual shapes are associated with predatory ant species. And as you get more generalist, it tends to converge on what we think of as that typical triangular mandible of an ant. So we sort of think of that triangular shape as the ground plan or the sort of basic point of departure. And around that, you have many different shapes that have evolved and often evolved to do different specialized functions. Everything from a giant fishhook to a vibratome. So these you may have heard about. These are leafcutter ants, and their mandibles have certain vibrations when they are cutting leaves that it helps cut, just like you've seen these electric saws so a leafcutter ant mandible is very optimized for cutting a leaf, as you would expect. Just another neat example. This is a very specialized ant mandible called Monomyrmex. They have these big sort of hook-like mandibles, and this is for going down a millipede and actually snipping off the hairs that are the defensive hairs of the millipedes. So very specialized mandible for capturing certain prey. And I'm not going to talk about all these today, but I'm going to go into more detail on one of my favorite type of mandibles, the trap jaw ants. So trap jaw ants are power amplified, and I'll explain what that is in a minute, but they've evolved in 4 different groups, at least 4 different groups in distantly related ant groups. And you know, when we say power amplified mandibles, what we're thinking of is a mechanism that can store energy and then release it very quickly. So biological muscle is limited by how fast the muscle fibers can contract, right? So if you want to do this, you know, there's only so fast I can do that. But one way to make it faster is to store that energy and release it very quickly. So the same way with a mousetrap, we're storing elastic energy and then releasing it very quickly. Or all over human engineering, such as a bow and arrow, you're storing energy that is produced slower by our muscle and then releasing that very quickly. So it's a way to get generate very high power or very quick energy release. So you've probably heard of the mantis shrimp. This is another example of power amplified mechanism. It's how shrimp generate extreme force with their appendage. And you know, it's kind of a funny aside. There was a vet, so Sheila Patek, who is a scientist working on the mantis shrimp, and Andy Suarez, my collaborator, was doing a postdoc at the same place, and they sort of bet. He sort of said, you know, you should look at trap jaw ants. I bet they're way faster. So they bet a case of beer on that and measured the trap jaw ant, and actually it was like several orders of magnitude faster than the shrimp. So so trap jaw ants have a mechanism in their jaws that is much like a mouse trap, where they will actually store energy, and then there's some kind of a latch mechanism that will cause the release of that energy extremely quickly. So I'll just show you what this looks like for one group, Odontomachus. And you know, of course, one way they use this is to actually catch prey. And I'll show you that later too. But they also found a secondary function for this, that they will use their mouths to as a means of locomotion, so for ballistic protection. So basically, if they want to escape really quickly, they can bite the ground and use that to catapult them out of the way. So I'll just show this video that was generated from this nice paper. So this is Odontomachus, and it's going to bite the side there. So it can catapult itself many different so it's going to bite. Got another one here. So this one will bite the ground and catapult it into the air. They're not very graceful flyers or graceful landers, as you will see, but they can shoot themselves many times their body length. And actually, if you've ever collected one of these, if you open the colony, they'll start popping around like popcorn. So, yeah. A little bit of hard way in there. Okay. So I'm going to talk about Strumigenes, which is one group, this clade of ants here. And you'll see that Strumigenes is one of the hyperdiverse genera of ants. So there's about 900 of described species and probably many more species that are undescribed. And a little bit of natural history about Strumigenes. As I mentioned, they are hyperdiverse. They're highest diversities in the tropics, but they do range up here. There are some species in this area. And they're all leaf litter predators. So they're sort of lions with a leaf litter going around and eating different prey. And they're also very small. You can see here how big they are compared to a finger. And many of them are predators of springtails. So springtails also have a spring-loaded escape mechanism. This is just a video of that. And so this is thought to be a mechanism that evolved to catch extremely fast escaping prey. watch till they land, but they fly off the screen.
They weren't graceful.
Yeah. So this is an example of Strumigenes hunting. You'll see it has the jaws wide open, 180 degrees, and when it detects prey, it slams the prey, stuns them. It will pick up the prey and move the gaster, the abdomen under, and sting the prey to immobilize it and then bring it back to the nest. But this is another Southeast Asian species that actually adopts more of a sit and wait strategy. So they'll basically open their mandibles in the lock position. And this is actually sped up 20 times. The speed is 20 times, and they're perfectly still. You'll see another ant kind of like come in here in a minute and see how fast they are. But they're incredibly good at being still, and they're very patient. So sometimes they will wait while prey crawls over them and gets right into the right spot before they will strike their mandibles. So this is actually a colony of Strumigenes rotegenes, and you can see many of the individuals across there that are in that sort of set trap position waiting for prey. Okay, so if we look in more detail, the trap jaw mandible. So this is from a CT scan. Of course, the yellow is the mandible, but this structure here, the green structure, is the labrum. So the labrum has sensory hairs underneath it. And when the mandible's open, it sort of slides in there and becomes a block on the closure of the mandible. So that, and then this red structure is the closing muscle of the mandible, so that will load the spring. And when the firing happens, when some prey will touch the sensory hairs, and a signal will be sent to these green muscles that pull the labrum out of the way, and that releases the mandibles to close extremely quickly. And there's all kinds of different changes in the head associated with this trap jaw, including special giant neurons that connect those sensory hairs to the muscle that releases the trigger. So basically, the reason it connects directly is because it is much faster to connect the sensor to the release than going through the brain, which slows down a lot. So many animals that have extremely fast reflexes, just like when you kind of do your knee like that, that doesn't go to your brain. That just automatically happens. And so that's also what happens inside this mandible. And you also see differences in the muscle structure and other morphological changes. But one reason that Strumigenes is interesting for evolution is that about a third of Strumigenes lack this mechanism. So there's variation in this genus, allowing us to look in more detail about how this evolved. So about a third have short mandibles. We sometimes call it a gripping mandible because they'll grab onto prey and sort of hold on for dear life and try to sting it, often while the springtail is kind of bouncing around and dragging them around. But the others have this long power amplified jaw. And this has been a sort of the evolutionary relationship between these forms has been a debate about as heated as ant taxonomy gets, I think, over the years. But it actually goes back to some really interesting papers by Bill Brown and E.O. Wilson from the 50s. And actually, one thing that's interesting is that E.O. Wilson did his master's thesis in Tennessee working on the behavior of these different forms of what are now Strumigenes, and started corresponding with Bill Brown, who was at Harvard. And actually, that's how E.O. Wilson ended up at Harvard, was discussions about this work. And actually, after he died, I got access to some of his old letters between Wilson and Brown, and where they're discussing all the ins and outs of these species. But also, I thought it was kind of cool because he's sort of writing like many of us have. Is there any funding or anything that we could do to come to your lab? So what do you think about my chances of getting a fellowship or an assistantship? I've got a good scholastic record, but I imagine I'll need a lot more than that. So many of us have written emails like that. Just remember that E.O. Wilson was there one day too. Okay, so and this project was a collaboration with many people, but I want to also highlight Doug Boer. I don't have his name up there yet, but Doug Boer, who was a grad student at the time, is now in Georgia, to reconstruct the evolution of this trap jaw mechanism around the globe. So we consolidated material from museum and personal collections around the world. We sequenced around 1,100 specimens from about 450 species, so a really broad phylogeny. And also we CT scanned and segmented to analyze variation across the phylogeny. And we did high-speed videography to measure the kinematics of the mandible across different forms. So to look at what we found in a nutshell, this is the broad sort of global phylogeny of Strumigenes, and the red color are all the trap jaw ants, and the gray color are all the ones that lack the trap jaw. So when you do ancestral reconstruction, you can find that the gripping mandible is ancestral, but this trap jaw has evolved independently in different parts of the world many times. In fact, the trap jaws that are in the neotropics or the Afrotropics or Asia, many of them used to be described in the same genus, but actually we found they evolved independently, and any morphological similarity is convergent. And that's actually why there was a big heated debate in taxonomy about these, because if you look at the trap mechanism, you'll group them one way, but many other characters were sort of giving conflicting signal, which makes sense now because there was this widespread convergent evolution. And I think if there's sort of one thing that I think we found doing all the molecular phylogenies in the last 20 years on ants is that there was a lot more geographic separation than we would have thought before, and a lot more convergent evolution across different forms, not just in trap ants, but across many different lineages. So this trap jaw mechanism evolved between 7 and 10 times independently around the world. Excuse me. But there was a one surprise is that this binary distinction between short and long mandibles was sort of a simplification. And we found that there was another short mandible form that actually had this trap jaw mechanism. It just has these sort of short mandibles with big teeth, kind of like a bear trap, and that wasn't actually recognized before as a trap jaw. And when you look at the evolution of the two forms, so the orange is the short trap jaw, you find that the long trap jaw mechanism is the red, has evolved 3 different times in different areas around the world. But the short trap jaw has evolved 3 to 7 times directly from that gripping mandible, and then 4 more times from shortening from the long trap jaw. So you see a shortening of the mandible. So it's a very complex history of these different forms. And I won't talk about this, but just to say there's many different evolutionary pathways that assembled the different species in different areas, but you always get the same collection of basically 3 ecomorphs in every area, even smaller islands, either through dispersal or evolution of one form to another. So when you look at the performance of this mandible, it's also very convergent. So you can see here, but the closing duration and the acceleration change by many orders of magnitude. That's a log scale, so many orders of magnitude difference. The mandible closes 5,000 times faster than the blink of an eye, so extremely fast, and also its highest acceleration of any resettable animal part that's ever been measured. So there may be some explosive cells and things like that that are faster, but anything that can reset, it's the fast, over 1 million Gs in acceleration. So really, really incredible speeds that can be generated. So whenever we find this kind of convergent evolution, we want to ask, well, did the independent evolution solve this problem the same way? When you look at that big phylogeny with the different distantly related trap jaws, they sort of achieved the same function with different designs. Different parts were modified into different parts. But within Strumigenes, and you can see each column here are sort of different lineages with similar phenotypes. We can look and say, are all the parts modified in the same way? And basically the answer is yes. I mean, you don't get really different design of trap jaws. All the same parts are modified into the trigger and the sensor and the block, compared to where they are elsewhere in the world. So we can ask, what about the architecture of these traits that facilitates this so-called deterministic evolution? Facilitates when we get evolution discovering the same designs over and over again. And previously, in other cases like C4 photosynthesis or mollusk eyes, you can find intermediate forms that sort of help you understand how one form could have evolved into another over time. So like as stepping stones. And when we look at Strumigenes, we can also see this, a real continuum of forms from the gripping mandible to the trapjaw mandible that we recognize. And I'll sort of show you a video that I think captures this really well. So this is the ancestral gripping mandible. This is the labrum. And this normally rotates out of the way before the mandibles close. And I want you to also notice that Strumigenes have this, what we call a basal process on the mandible. It's this sort of little nubbin that is there. Not all ants have that. And normally the mandible, the labrum will move out of the way and the mandible closes. But one thing we found was actually a sort of primitive trap jaw that is only barely modified from the gripping mandible. You can see here the labrum gets these little pockets on the side, and the basal process elongates, and now it catches on the labrum when it closes and changes the function. So when it's open, it's only basically parallel. But you can see there that was the key change, and it changed the function of the mandible. So now the labrum muscle is what's releasing that energy and closing the mandible. And from there, we can start to see a real reorganization of the head. So the basal processes migrate back. This is a 90-degree version, and then it'll go to a 180-degree version. You'll lose the dentition on the mandible. You can see it open 180 degrees. And finally, you'll see, well, not finally, but you'll see a 270-degree version. That was the biggest opening gate that we found. And then in some groups, you start to see a shortening of the mandible. So this is a short trap jaw that opens 180 degrees. And finally down to back to a 90-degree opening of the short trap jaw. So from that, we can see not that evolution necessarily took all those steps, but you can now see how you get this more complex mechanism evolving from something simpler. You can only very small realignments can change the function of the mandible, and then after that you get this whole evolution of the head to optimize around that trap jaw mechanism. Okay, so yeah, so the three keys here are consistent ecological selection for speed. So the non-trap jaw actually is the fastest ant mandible that's been measured that doesn't have a power amplified mechanism. So there clearly was some selection to try to be fast. You had that pre-adaptation of the basal process that sort of was ripe to be modified. And you have a nearly continuous pathway of stepping stones between the different phenotypes. So just to mention, we have also ongoing work looking at the genomics and developmental mechanisms to try to understand in a deeper way how that convergent evolution happened. I won't go into all that. But I want to move on and tell you about another species that we studied, and this is a wood-borne species called Melissa tarsus. And this is a very special species. It has many what you would call innovations that don't occur in other ants. It actually lives all of its life inside the galleries it chews in living wood. So it's the only ant that can chew and bite through living wood. Other ant you see in wood, it's always in dead wood. And inside those galleries, it tends scale insects and eats the shield off the scale insects as it's shed. And it has special, modifications of the mandible and the head that facilitate that chewing mechanism. And we did study that. But it also, the most notable thing is it has these really interesting legs. So this isn't because I just mounted it improperly here. Those middle legs actually are oriented to the top, and it actually will walk on the top and bottom of the tunnel at the same time as it's moving. And it's something like this you can see here. And it's actually so derived that those ants, if you take them out of the tunnel and put it on a 2D surface, it can no longer walk. So this is a video of what it looks like. It just kind of flails around. We put it back in after. And just, to tell you a little bit about what, why we think that is, it doesn't need to do that to really walk through a tunnel, because many ants walk through tunnel, but it has these modified legs that actually we think brace in the tunnel. So if you want to chew through something really hard, you can have powerful mandibles, but you need to have leverage or else you'll just push yourself back, right? So we think it uses that to brace inside the tunnel. The reason I mention it is in studying this mandible, we, or really my colleague Roberta Keller, sort of had some insights about how kind of normal mandibles work, and that actually that ground plan mandible is not as boring as we may have thought it was in ants. That actually ants have a very unique design of their mandibles compared to other insects. And yeah, so I just want to mention this is the work of Roberto Keller and postdoc Dr. Hita Garcia and my student Julian. So just some background, many of you know, but one of the ancestor state in insects is monocondylic mandibles. And when I say a condyle here, a condyle is the ball in a ball and socket joint. And so the ancestral mandibles are monocondylic. They have one ball and socket joint for each mandible. And that is a lot more flexible, but also less stabilized. And one of the original innovations in insects was the evolution of a second joint that stabilized the mandibles and so that they open and close a bit more like a door, right? And that was thought to be one of the key innovations in insects. But one observation is that if you look at ants, ants have a large gait. And we can zoom in on this. I mean, look at the angle of that mandible as it's open, okay? It opens very far. And actually what Roberto noticed in studying Melissa tarsus is that ants have a modified dorsal condyle. So here is a wasp. You have a condyle at the ventral and the dorsal side. But ants have a modified condyle on the dorsal side. So you can see better here with the mandible taken off. It has this long elongated groove instead of a ball and socket. And actually many ants, if you look at them, they have this sort of bald patch on the outside. And we always, people use that for identification, but we didn't really kind of know what that was about. And the reason is when the mandible opens, it sort of pops out and articulates with the outside of the head capsule. And that's why that structure there, you kind of pairs them. So showing this another way, it looks like this. When the mandible opens, it pops out of the joint on one side. And the consequence of this is that ground plant ant mandibles have a twist when they open. So they actually twist as they're opening. And it's a more complex motion than is possible with typical dicondylic mandibles. So another example of what this is actually, most insects have a very sort of door-like opening of their mandibles, but ants move in this irregular way as they open. And, we don't, I guess, have really solid evidence for this yet, but what we think this is about is that twisting allows for a single shape to be a multifunctional tool. So if you want to cut something, you want to have something that looks like a pair of scissors, right? That has the sort of long axis perpendicular or aligned with your axis of motion. But if you want to carry something, you don't want to carry something with a pair of scissors, right? You want to do this. And so actually that same mandible can cut and then open and actually grab on and carry things. That's what we think. If you have any other ideas, let me know. So the significance, well, ants are pretty good at carrying stuff. You may know that. And they're some of the only Hymenoptera that actually can pick up their own brood and move it to a new location and move their whole nest. So that's a really important innovation for ants. And so we had this hypothesis that maybe the diversity of all these different forms could relate to the diversification of the motion as well. So actually that having that joint that they can play with allows them to have a different diversity of motions. So what we did is we did a kind of motion capture where we CT scanned mandibles in the open and closed position and then calculated the difference and how that mandible moved. And they compared that to a bunch of other insects, both ants with kind of normal ground plant mandibles, and also weird, I'm just calling them weird mandibles, all these different shapes, and also other non-ant insects. And I'm not going to go into too much details, but basically what we're measuring here is how much that axis through the condyles moves. So how much that top joint moves out of where it was originally. And you can see that sort of non-ants are all down. So this axis is ventral movement, this is dorsal movement of the mandible. They're all sort of down in this area. Ground plant ants have sort of higher dorsal movement, but also these weird ants are kind of all over the place. So they're really going in different types of movements, and also some of them look more like other insects, like a very regular movement. But I think we do have some evidence that this diversification of motions accompanies that diversification of shape. It's something we're looking into, and we're doing other kinds of videos and things along with that. But I want to also, talk about one more thing, and that is what I said in the beginning is what we want to do is not just study some species that have cool traits, but actually generate really big data that captures the diversity of a whole group. And for phenotypes, it's always been challenging because, as any of you know, micro-CT is pretty low throughput, and also it's hard to get access to the scanner, it's expensive. But there are some ways to generate big phenotype data, and actually it's easier with insects than it is for vertebrates and some of these other groups. And so this is why we've been working with our colleague Thomas van der Kamp, who is at a synchrotron facility in Germany on high throughput synchrotron imaging. So a synchrotron is a particle accelerator. It has X-rays coming off of it, and you can put a CT scan there. And his group has actually built a robotic pipeline to scan insects in a high throughput way. So these are all specimens and vials that have been loaded up. And so we'll make these big boxes of specimens. Robotic arm will move it into the beam. And just because synchrotron X-ray beams have different properties than our lab CT scanners, what takes us about 12 hours in scan time to get a really good image for something the size of an ant, you can do it in the synchrotron in 30 seconds. So if you load many of these specimens, you can do hundreds and thousands of specimens fairly rapidly. So we did a project we called AntScan, and actually the Smithsonian and Ant Lab was a participant in this, and we got our collaborators from around the world to send us specimens from all over. We databased them and loaded them in files like this. And here you can see our team in our lab in Japan. And this was kind of a COVID project. It got a lot of different twists and turns, but my student was from Germany, was able to go there in the pandemic and do scanning. And we were able to scan 2,200 specimens in 1 week. So by far more scans that have ever been done by all labs, in working on ant diversity, including our lab, who's been scanning ants for 10 years. We were able to scan, 10 times that in a week. So we've got about 850 species, 210 genera. You can see this is a genus tree of ants, and all the red are genera that were included in our first round of scanning. Different casts, and we also coordinated with genome sequencing projects, and 238 of the specimens were from the same collection that were used in big genome sequencing projects, so that we have a genome and a phenotype to actually do different analyses. And just to show you the kind of information you can get off one of these scans, so this is a specimen that we've now rearranged, posed, and segmented, so gone in and painted the different parts. And you can see, you can get all kinds of musculature, the nervous system, the reproductive system in the abdomen, the digestive system, sting apparatus, lots of information in there to do all kinds of different comparative studies. And just to mention, it's great to say you have 2,000 CT scans, but it's really hard to access that information because even to sit down and open, each scan is about 10 gigabytes of data. Sit there and open it would take you about a year just to open and close each file. So we have to make algorithms that actually can go in for us and recognize different parts that we're interested in. In this case, one of my students made a brain detector and can pull out that brain. And so this is sort of a big area of research now, is trying to develop computational methods to digest all this image data. Okay, so I want to end on something a little more fun, and when you work on 3D data, it provides a lot of different endpoints of places it can go. It's not something we were really thinking about that much when we started, but I just want to show you some of the different uses of this kind of data and how it can change how we interact with biodiversity and interact with collections. So one thing is online galleries. So we have a gallery on this website, Sketchfab, where you can go in. There's thousands of models that you can go and interact with 3D models. And actually, we have quite a lot of people following us there, and almost none of them are biologists. The people who are the most interested are actually 3D animators and artists, because if you look at the sort of level of detail you get off these kinds of models, it's far more detailed than, say, what you use in Pixar or one of these. So the people working for some of these animating studios are really interested, and they're often taking bits of different animals and put it together to make some kind of movie monster. You've probably seen that before. So this has been on the side, but we tried to kind of facilitate this for different artists who contacted us. Also 3D printing, of course, I mean, especially for outreach, and people just really like to interact with 3D models when you print them out and take them to different areas. Virtual reality, so I'm going to show you a model that was loaded into a okay. I guess it's not going to work, but
There's codec unavailable on that one, so I'm afraid that one might not play. Sorry about that.
that's sorry. Okay, yeah. So you can load a model in virtual reality. What you would see is a biologist, or one of my colleagues, looking through 3D glasses and interacting with the specimen of basically what be in their full environment, the size of a horse. You can look at a specimen. I can tell you, it really changes your perspective on the insights you have when you can see a specimen that size in your own sort of 3D environment. I mean, it kind of accesses different parts of your brain about intuiting, what these different structures are used for. Another thing we have been experimenting with is augmented reality. Let's see if this will work. Yeah, okay. So one thing we did was we described some species, and actually we wanted to see, well, what can we do with augmented reality to enhance those descriptions? So we actually programmed built a augmented reality app for the iPhone just to go with one taxonomic description. So we hired some developers to do this. And one story, we originally named all the species after Pokemon, from Pokemon Go. And we wrote this really beautiful letter in Japanese to the inventor of Pokemon, which you may know, the Japanese person who grew up liking insects and collecting insects, and that was inspired Pokemon. And we got a letter back from their lawyers saying, do not do that. So, I don't know if it ever got to them, but so we ended up naming them after the movie Ready Player One, you may know, and we didn't ask permission, we just did it. So but what you see here is 3D figures. So if you hold the app over the paper, the figures will pop out and you can put the specimen anywhere. And there's also 3D, you can see the distribution map in 3D. So yeah. And this sort of leads to my last point. In this movie, Ready Player One, people are going to spend a lot of their life in these sort of artificial 3D or environments. And it's a bit dystopian if you think about real biodiversity in a way, but also it's kind of an opportunity. And one thing I'm motivated is how can we get real biodiversity into this digital realm and these digital worlds? And that's kind of what I'm obsessed with a little bit is making sure that it's not just made up monsters, that this can be a new way for people to actually interact with biodiversity. And just to show you the kind of things you can do, so this is, I hope this works, yes, okay. So this is an animation made from those X-ray scans from AntScan. We colorized and posed it and given some motion. So these are morphologically perfect models, you know, basically from the real specimens. And so from that, you really see, you know, this is one way we can sort of populate the digital world with real insects and real biodiversity. Okay, yeah, so I just said, I mean, imagine we have all these different data sets, including geographic distributions. It really gives us some new opportunities for using that data. And with that, I'm just going to thank you and thank all the collaborators in all kinds of different parts of this talk. So thanks.
Right. That was an excellent talk. So I'll go ahead and open up the floor for questions. Anybody have questions? Let's maybe start with the real world first and then move into Zoom land.
Movies out of that.
Yeah.
Cartoons and then build it up to a regular movie.
Yeah, yeah. So that's one thing that I've thought about, and I'm trying to, you know, like I said, it's been a bit haphazard interacting with these artists, but I would love to find a more organ. Usually it's them saying, "Can you send me this model?" And we're like, "Oh, too much time," you know. And then they bug us a few times, and we finally send. But I think one thing I would like to do, I figure out how to do it, is, you know, have some better point of contact between people who work in the Hollywood industry and people who work on, and us, basically, people who work on organisms, to actually have that cross-fertilization a bit better. And another thing is, like, well, I showed these videos of these ants that we kind of brought to life, but that took incredible amount of time to do that with a couple specimens. But one thing I'm starting to try to get some computer science students at Maryland to do is see if we can use AI to do that more automatically. You know, can you use, so then we'd really be in business. You could open a digital museum with, you know, realistic looking moving specimens across huge numbers of species. Yeah.
What's the relative speed of the trapjaw ants jaw closing to the Columbola springtail fire?
Yeah,
how much
so
faster?
The Columbola is very fast, but the trapjaw is still about 2 orders of magnitude faster. Yeah.
At a much larger scale, with respect to trapjaw evolution, do you find other similar mechanisms at a higher level? Like, why don't we see lots of other insects with trapjaws? It seems like a formicidal kind of thing.
Well, there are trapjaw spiders. I think someone said there. You know, so Hannah Wood here has worked on power. So there are different insects with power amplification mechanisms of different kinds. So sometimes jumping mechanisms, or like the Fundra or the chelicerae, you know, have a certain mechanism. But it isn't that common. Yeah, I guess it's hard to answer, right? Like, why aren't there more? I mean, one thing I will say is, across all ants that I'm aware, once the trapjaw has evolved, we've never had a case where it was lost. So it clearly is a pretty good thing to have. But yeah, I don't know. So I think we haven't looked in detail at these other genera, like what if we can figure out. One problem with the other trapjaw genera is there aren't intermediate forms like there are in Strumigeni, so we couldn't really reconstruct easily the stepping stones. But,
I'm wondering if it's just the head is well adapted for powering mandibles to pick stuff up. That's a common prognathous ant thing. I wonder if that has some sort of.
So basically, another way to ask is, like, why don't all hens have that?
Yeah. Yeah.
Good question. I think we need to learn more about the trade-offs, I guess, involved. Yeah. Because it's really cool. You know, there probably are special investment you need to do to have on the materials level, and there's huge kind of physical forces that go through those things. But yeah, it's a good question.
Do any of this Rumigenes have like different casts, like leaf cutter ants do? And like are the reproductive forms, do they have different kind of?
Yeah, so the queens have the trap jaw mandible, the males do not. So the males have reduced mandibles like many male. And there are most species of ants, Strumigenes do not have polymorphic worker casts, but there are a few that have something like, I mean, it's not quite like a soldier, like a Fidoli that has really different allometry to it, but there are some different size variations in some of them. Yeah.
This may be not of general interest, but I'm wondering what you think about the developmental origin of the basal process in Strumigenes manifolds. Is it just another tooth, or is it something else developmentally?
Yeah, so one thing that if you call that a tooth, and morphologists will yell at you, no, that's a basal process. But I actually don't know about the homology of that. I don't know if you have thoughts on that, but you know, I am gearing up now. It's one of the projects we're really starting is to go deep on the developmental biology of all of these traits in that trap jaw, working with an actual developmental biologist. But yeah, that is something I would like to look, that would be cool to look at on a broader scale and see who has that process or what that's doing. Yeah, I guess that's all I can say. It's a good point. Yeah. Oh yeah.
I have a question out here in Zoom land if there's nobody with a burning hand down there in the real world. I'm very curious about the kind of the functional mechanics, but especially the material characteristics of those of those cuticular structures that are being placed under the most stress in those trap jaw mechanisms. Have you observed any kind of modification to the structure there, or of the mechanical properties of the actual, of the actual components of the cuticle, or the different layer types?
I can say that the labrum, so that structure that becomes the block, that is much more sclerotized in the trap jaws than it is in normal Strumigenes. But I think it is very likely that the actual cuticle has different mechanical properties than normal cuticle. But as far as I know, no one has actually looked at that. My colleague I mentioned before, Andy Suarez, there's some engineering labs there that have been looking at this more from an engineering perspective. I don't know where they are with that now, or if they've looked at that, but I think they've measured some of the like Young's modulus and some of the material properties. But as far as I know, no one has conclusively shown that it's different. I will say that, you know, we have another study looking at evolution of cuticle thickness. Strumigenes is a bit thicker than it should be, basically based on its phylogenetic position. But I think there probably is some mechanical, robustness for mechanical stress. But one thing I didn't mention is we actually don't know where the spring is in that. So we don't know what structure is storing the elastic energy. There isn't like a very specialized thing. It's probably deformation of the whole head under stress, but we don't know that for sure.
Interesting.
When you do the microCTs of the activated mandibula and then deactivated and you compare the angles, you see some deformation of the head or?
Well, you mean the for the other project where we? Yeah. So in that case, it's not under stress in the same way, because in order to move the dead, manipulate the dead mandibles, you have to kind of cut the apodemes. So it's not under stress in that way. What that does is kind of tells you where how the cuticle would guide the mandible as it opens. So that wouldn't be able, you wouldn't be able to do that. But you could, I mean, in fact, I know Andy had done, at least with maybe one of the snap giants, so there's another type of ant that instead of a trap that closes, they push against and then do like that, and it's like a weapon, and it smacks something on the other side. And he did some things where they like bent it and saw how strong it was for breakage and things like that. So they are, I think, but yeah, that hasn't been something that I myself have. Anything else in Zoom land there? Yeah.
Those ants that do living wood, chew through living wood, is that the situation where a tree drops a limb? And that would be.
You mean the cause of it dropping a limb?
Yes, right.
I don't think so. They are more like living under the bark of living trees. From what I know, they're only found. Those ones are only found in Africa, actually, the Afrotropics. So.
Oh, really?
Yeah, but they're quite common. But another interesting thing about those, they have many different innovations that other ants don't have. They're the only ant that the adult worker produces silk, like they use silk to build. They have different enzymes, all kinds of other adaptations for chewing wood and living there. They're the only ant species that raises herds insects only for meat, basically. They don't get they do out of that. But but interestingly, there may only be one species in that group. Maybe it's a complex of a couple species, but a lot of innovation packed into one lineage. Not to get these generate ants that have 1,000 species and not as many different interesting innovations.
And they're limited to Africa.
Tarsus is Afrotropic. There is another sister genus that has some of those adaptations that lives in Southeast Asia. It doesn't have the modified legs, but it lives in raises scale. Okay.
Yeah, just say early on there's ants that prey only on millipedes.
Yeah, actually a lot of ants prey only on millipedes. Surprising number. Yeah.
They're common enough that they have it.
Oh yeah, they're like the Big Mac of the leaflet.
Any questions in the chat?
No, I don't have any questions in the chat. Lots of comments on very cool and engaging and fantastic talk. So I guess just kind of one more time, does anybody have questions either in reality or in virtual reality?
Just a comment. I think that have this morphological, I think it's amazing. But so I think that for understand function, also we need the same thing with behavior. So how we can, so we have a lot of information in morphology, but so to do the 3D animations and to understand the complex muscular activation, how can we reach that level with the behavioral part?
Yeah.
So to that.
Yeah, I mean, I think I think that's a step ahead a bit because we're just now trying to deal with how do we even analyze that much data just to measure head width or something, off that many scans. But yeah, I mean, I think, of course, like the videos I showed you, we just kind of wiggled it around. Maybe they have different stereotypical movements. I mean, you could imagine a world in which people had videos of ants, and then you could grab off that motions and movements, and then use the model and animate that in the model. I mean, that's technically possible, but video and ants is one problem we've had is that they're kind of small, and so it's not easy to get the rig to get a good video of ants moving, for just an average person out there. And but maybe I think the new iPhone has like really better a macro camera. I don't know if anyone has those tried it, but maybe in the future that'll be more possible. But
in general, the behavior is well studied, but how much of this is more speculative or
about the trap jaw
about the behavior, the feeding behavior?
So for the trap jaw ants, I mean those, that's been studied. And that's the kind of thing they were doing. E.O. Wilson was doing. He was saying, well, what kind of jaws? What do they eat? And all this sort of things. Now the problem there is, over the years, and we got out every study. Someone wrote the feeding habits of something in Malaysia, and there's like a handful of species around the world that have been studied in that way. But that once you have the biology and everything else, that's like gold to get these kind of studies to understand. But so maybe we've studied 10 species and there's another 900 that haven't been studied. But one thing I will say is that, I talked about the kind of, sorry, I'm losing my voice, the dominant different forms, but there's actually also a lot of really weird morphologies out there in this genus that have really upturned mandibles or that are more rare. And to the extent they've been studied, sometimes they will specialize on either a unique hunting tactic that the others don't. So one will actually be in tunnels and kind of bury itself, and then the mandibles will, any kind of long arthropod that's going through the ground, it'll kind of bite it from below and catch it. So that's one. Another, well, there are some that like specialize on Dipplura, and they have others. Then there's some other forms that evolve convergently in different areas, but we don't know what they're for, And so it is going to be a problem to get that natural history for all those species. And one of my postdocs, the guy who made the videos of the ones that are staying really still, he's in Japan. He's working on trying to get more natural history data, but you still can only do a handful of species really to study there. So that is, a problem for the kind of, I guess it's easier to get big geographic data and morphology data, things that you can get from a collection, basically, than it is to get something where you need to go in the field in Malaysia to get that data. Yeah, some of these ones, those like short trap jaws, we've never seen them. People have never seen them alive and studied them. They come up with Winkler samples in Costa Rica, and no one's ever made field notes on what they're doing.
Are there any issues with shipping the samples internationally for like the answers project?
Yeah, I mean, so in terms of legal, what we did was we had a few partners and we made MOUs or did whatever we needed to do for the Nagoya protocol and all that.
Because we had a speaker 2 times ago that was talking about the illegal insect trade and especially ants. But that's of course more concerning live specimens, but it's interesting.
So generally, I mean, from a control point of view, in terms of like, people don't so much care if you send ethanol preserved specimens. Right. From a Nagoya protocol point of view, there is a certain countries you need agreements. Like with Brazil, we had an MOU, but one thing I want to mention about the project I didn't say is that data is all going to be like from the get-go will all be online, for everyone to download and use, so that now you can, if you want to do a PhD on the evolution of something in ants, you can basically do it from the scans without having to go and rescan all that stuff. So that has been a big problem with micro CT studies. It's because it's so expensive to get them and so hard to get access. It's just people can't do it in most places. So that was one big motivation for this project, which is good for me because I just moved to Maryland and we don't have one. So I guess you all have one here. Great. But yeah, so I think that was one reason people were keen to send us specimens, because then they have all the scans and they can work on their own faunas. But it is an issue with we need to be careful much more, and even when I was started 20, 30 years ago, people just sending everything everywhere and I didn't think twice. Okay.
If there are no more questions, thank you very much, Dr. Economo. Much appreciated. That was an excellent, excellent presentation. All right. Are there any further items of business or announcements? Any of the officers or members of the general public at this time? All right, hearing none, I am adjourning this meeting of the 1250th session of the Entomological Society of Washington. Thank you all for joining. It's been wonderful having you. Have a fantastic evening. Thank you.